Alright. Hello class, so again my name is Andrew Menezes and today I'm going to be talking about utilizing benthic macroinvertebrate data for assessing water quality and ecological health in freshwater environments. So here's a look at what I'll be covering today and we'll get right into it. So benthic macroinvertebrates, or benthos, are bottom-dwelling aquatic invertebrate organisms that live among stones, logs, sediment, and aquatic plants in various surface water bodies. They are large enough to see with the naked eye, hence macro, and are caught with nets containing a 500 micrometer mesh size. Benthos include animals such as insects, worms, mollusks, crustaceans, and mites, which can be used as biological indicators in assessing water quality and ecological health. So what makes benthos such ideal indicators? Well, according to the literature, this is because they are sedentary and can thus integrate site-specific impacts. They are ubiquitous and are easily collected. They are abundant and exist in all surface water bodies. They are diverse and respond to a wide range of stressors. They are an integral part of the food web. They are relatively long-lived and can reflect cumulative impacts. And finally, they are relatively inexpensive and do not require special permissions to sample, unlike fish. Now, in order to interpret the data presented by benthos, several different metrics and indices have been created. These metrics are based on characteristics of the biological community that change in some predictable way as stressors arise or human disturbances increase. Different metrics are utilized depending on the purpose of the assessment, and these may incorporate different organism pollution tolerances. For example, mayflies, stoneflies, and caddisflies, also referred to as EPT taxa, typically require higher oxygen levels and are associated with clean or unpolluted water, while worms, leeches, and midges typically do not require oxygen in high levels and are thus associated with polluted water. So an example of one of these metrics is the EPT taxa metric, which summarizes the taxa richness within pollution-sensitive insect orders, which generally increase with increasing water quality. So those guys at the top there. However, in general, diverse, well-balanced communities reflect good water quality and aquatic ecological health, while communities dominated by relatively few taxa suggest some environmental stress. Now, along with the metrics, several different protocols have been developed for benthos studies. These protocols include the collection, management, assessment, and distribution of information on the biological condition of aquatic ecosystems. The Canadian Aquatic Biomonitoring Network is the nationally standardized program with protocols used for benthos studies in Canada. CABIN was developed and is maintained by Environment and Climate Change Canada, and it was done to provide participants with the standardized protocols for the collection of benthos and stream information that uses the reference condition approach. Now, with the reference condition approach, the study design begins with the identification of a test site, which is typically a priority area or region of concern based on hydrological or biogeographic boundaries. Reference sites are selected where environmental attributes are similar to the test site but are minimally impacted by anthropogenic forces. A bioassessment model is developed from the reference site data, which is used to define the normal range of biological communities that should be found at the test site if minimally impacted. The test sites are then compared to the reference sites using the bioassessment model, where divergences between the benthos communities at the test sites and the reference sites are compared in order to assess the extent of potential impairment to water quality and or ecological health. Now, there are several advantages to using benthos in this matter. For one, sampling benthos can be more cost and time effective than traditional sampling, as there's the ability to measure cumulative impacts in the environment, including impacts from contaminant pulses and chemical interactions. Also, the composition of the benthos community can be useful in narrowing the scope of chemical sampling in some situations, so for reconnaissance. As well, other stressors such as the presence of exotic species, habitat degradation, and fluctuations in water quantity can also be captured while sampling for benthos. Benthos data is also helpful for food web interactions, and lastly, the information derived can be shared across networks using CABIN tools and databases. However, utilizing benthos data does have some limitations. For one, some metrics or protocols require taxonomic identification beyond the order level, which requires very specialized training and can be very time-consuming. Secondly, it's difficult to distinguish the effects of multiple stressors using benthos data due to the web of indirect effects and interactions between ultimate causes of ecosystem degradation and immediate stressors in the community. And lastly, the reference condition approach may not be possible or ideal when there is a lack of appropriate reference sites to compare the test site to. This is common in areas that have long been settled and used by humans, such as southern Quebec. So in summary, as has been seen in this presentation, benthic macroinvertebrates are ideal biological indicators that present data that can be quantified and modeled in order to assess the condition of water quality and ecological health in freshwater environments. So here are my references. I'd also like to acknowledge and give special thanks to Dr. Nagati, the TA, and my primary and secondary reviewers. And thank you. what was it again? 2?